Normalization of RNA-seq data is a critical pre-processing step which can make or break downstream analysis. There have been lots of work in developing new and improved normalizers for RNA-seq data, but almost none focusing on how to measure the performance of those. Most of the criteria being used in the literature by far are subjected to biases, biomodeling assumptions, or optimization objectives. Here we propose a new way to measure normalization success, which is called condition number based deviation, or cdev for short. cdev measures the dissimilarity between two expression matrices, and by measuring against an experimental route truth, cdev can quantify normalization success without bias, either for or against any modeling assumption. We use two types of route truths to demonstrate the characteristic of cdev: simulated and experimental. In both cases, a route truth normalization is obtained by scaling its sample by the count of two reference genes. In simulation, those reference genes are simply non-differential ones, and in experimental datasets, they are ERCC spikins, which are a synthetic RNA sequence, and were added to on sample at the same concentration. Here is the collection of on dataset with ERCC spiking that we have collected and reprocessed. They are now all available as our data packages here at this repository. Now, in order to compute cdev between two expression matrices x and y, we suppose x can be transformed into y by the matrix B. Then cdev between x and y is the ratio between the largest and the smallest singular value of B, which is the condition number of the matrix B. With this definition, cdev is guaranteed to exist, so it can always be computed for any two expression matrices of the same shape. And when the transformation only involves scaling the sample, it is also conveniently symmetric. And most importantly, cdev is scale agnostic, which means that the cdev between x and its multiple x prime is always 1. And thanks to this property, cdev would measure the differences in expression pattern rather than the absolute expression levels. We found that cdev is highly indicative of normalization quality. This graph over here shows that cdev is correlated with both upstream and downstream indicator of normalization success. And these relationships are independent of the number of genes or the fraction of differential genes. Comparing with Fontain's MSE, a simple metric defined for two conditions, cdev is completely concordant on simulation and simple dataset. On complex datasets, like those with multifactorial experimental design, cdev is generally more robust than MSE. Finally, we illustrated the use of cdev by comparing several normalizers on both simulation and experimental dataset. We found that most of the time, none of the normalizers were able to obtain satisfactory result. In such cases, the true reference genes, those that are visualized here as the heat maps, were not stabilized after normalization, in contrast to the route truth. And these observations are properly quantified by cdev.